Mechanism of muscle fiber contraction. Objective: to study the sliding filament model of muscle contraction. The chemical processes involved in muscle contraction. In the human body, the muscular system consists of numerous muscles. Each muscle consists of bundles of muscle fibers, and each fiber is composed of numerous cylindrical subunits called myofibrils. A closer look at the sectional view of the myofibril shows the presence of myofilaments, which are of two types, namely actin and myosin. The myosin filament is thick and is made up of bundles of flexible myosin molecules, each having a rounded head which points outwards. The actin filament is thin and consists of two strands of actin protein molecules loosely wrapped around each other. Let us now observe the process of myofilament contraction. During the process of muscle contraction, the head of the myosin moves and attaches itself to the actin filament. The myosin bends backwards and the actin is pulled in towards the center. As a result, the actin filament slides past the myosin filament. One after another, the myosin heads attach to the actin filament, and the actin filament keeps sliding on the myosin filament. This is the sliding filament model. With the actin filaments being pulled inwards in one direction, the ends of the sarcomere are pulled closer, thus shortening the muscle fiber. To understand the chemical process of the muscle contraction, we will focus on a myosin filament. The heads of the myosin filament attach to the actin, but the attachment sites on the actin are blocked by rod-shaped proteins called tropomyosin. Tropomyosin shows the presence of another protein known as the troponin. To initiate the process of contraction, the calcium ions bind to the troponin molecule. This forms the calcium troponin tropomyosin complex and causes the complex to shift away from the attachment sites of the actin molecules. The ATP molecule binds to the myosin head. The hydrolysis of this ATP gives energy to the myosin head to attach itself to the active site of the actin filament. Finally, the myosin pulls the actin filament and ADP and phosphate molecule are released. Another ATP molecule attaches to the myosin head and results in its dissociation from the actin filament. The calcium ions are released, and the tropomyosin returns to its normal position, where it again blocks the attachment sites of the actin myofibrils. Thus, the myofibril regains its relaxed position. To recap the process of myofilament contraction, we see the calcium ions bind to the troponin molecules, thus forming the calcium troponin tropomyosin complex, which opens up the active sites on the actin filament. Then, the hydrolysis of ATP brings about the attachment of the myosin head to the active site of the actin. Finally, the myosin heads pull the actin filament inwards, thus causing the myofilament to contract.